So hello, everyone. Today we have Gaby Lohman here. I can say a few words about Gaby. She is a group leader at the Max Planck Institute in Tübingen. She is a developer of MRI and fMRI analysis tools and the main developer and maintainer of Lipsia, I believe, which in my circles is mostly used and famous for the kind of denoising, for example, wavelet denoising, other denoisings as well, also in combination with then corresponding statistics, which also gave you the Nature Methods paper not too long ago, I believe. She was acquainted with the layer fMRI field since the beginning. So I think in 2011 or when it was, you were involved in the mental imagery of the tapping with Robert Rampel, which was also the time when you were the formal main supervisor of my master thesis, and I was struggling with VASO and the bold condemnation therein. I think Gaby is most famous for, maybe for, or known for also the criticism of DCM. Today she's going to talk about Cylarim, which is a tool for layer fMRI folks, I believe specifically suited for ROI selection. Please, Gaby.
Yeah, thanks, Renzo. It's a pleasure to give a talk here in your meeting. And yeah, let's dive right into it. So there are plenty of challenges in laminar fMRI. Spatial resolution, of course, is one of them. And an obvious solution is to go for higher field strength, 7 Tesla plus something. Then, of course, we have the draining veins issue, the vasculature, and there are several solutions that have been proposed. For instance, use better sequences like BSSFP or VASO. Perhaps if we know a convolution kernel, we can also do a deconvolution and use activation contrast, so not baseline contrast, hoping that the bias involved in the draining veins cancels out if we subtract contrasts. Another issue is, of course, segmentation accuracy. We need to have extremely good segmentations of gray and white matter in order to be successful here. And for that, better algorithms would be a obvious solution, perhaps based on artificial intelligence. But then there's another issue, which is dependence on a good selection of a region of interest. And that, I think, is a bit under-researched, and that's exactly what I would like to address today in this talk. So it's really about how do we select regions of interest, or maybe how can we even avoid having to select a region of interest at all. So here's an illustration of what it means if you do not select the best region of interest. So currently, most laminar fMRI approaches do require the definition of a ROI. And here you can see these green rectangles illustrating different choices of an ROI. And obviously you would get very different results depending on where exactly you put the ROI. So there is clearly a bias in how you define the ROI. So what I would really like to have is something like what I'm showing here. So mapping that doesn't require any ROI at all, but something that tells me which parts of the cortex actually show, for instance, middle layer activation that is stronger than the deep layer or the superficial layer. So the image I'm showing here would be kind of a final result. And this was actually stolen from the Laney website. I will make heavy reference to Laney throughout this talk. So thanks, Renzo, for providing and maintaining Laney. And I guess there are also other people involved in the Laney website. So for this purpose, I'm now proposing a new software system called Cylarim for laminar mapping. The idea is really to get an entire map that doesn't depend on a region of interest. So let's talk about the common processing pipeline that people typically use. So obviously we need a gray-white matter segmentation, also CSF segmentation. We also need, in this case, an upsampling, for instance, to 0.2 cubic millimeters, something around that range. Then Leni, of course, is a prominent tool. And then people typically define a region of interest and then do their statistics within this region of interest. And obviously, these last 2 points are the ones that I would like to address and maybe avoid. But let's start with the gray-white matter segmentation. So here's a gray-white matter segmentation that was performed using a new tool that my PhD student Julius Steiglechner is in the process of putting out. It's based on artificial intelligence and it works really nicely. But obviously, for the purpose of layer segmentation, there's something missing. And that's what we can see here when I uh, overlay the blue lines here. So we do not really, in this, even though this segmentation is highly precise, it doesn't really distinguish between the two banks, the opposite banks in a gyrus. And that, of course, would screw up any kind of layer segmentation. So how does that work? So I'm now, so one of the first step is to propose an automatic post hoc correction algorithm that would deal exactly with this problem. So we start with an upsampled T1 weighted image, like you can see here. So it's back of the brain, somewhere in the visual cortex. The next step is what is called grayscale morphological closing. So I'm not sure if you're familiar with morphological filters. It's kind of an old technique, was invented like 30 or even 40 years ago or something. And there's binary morphology and also grayscale morphology. I'm using what's called a morphological closing filter. And I guess you can kind of see what it does. It kind of fills out the darker spots. You have to select a kind of a structuring element, kind of a spherical element, and then it move it across the image, and it produces an output like you can see here. So let me go back. This was the original image. This is the closed image. And then we subtract. And as you can see, this difference image shows these uh, small parts, the CSF-filled parts in the interior of the gyri. And then we do a thresholding, and we do what is called 3D skeletonization. And now we have really uh, the middle of the of the gyri, and we can use that to actually, update the segmentations. So here, a couple of slices of this image. This was the original segmentation, and here we see the post hoc correction. It looks like it's not complete, these blue lines here, but they actually are. It's kind of difficult to visualize because we are in 3D and it's also upsampled to zero point 0.2 millimeter. So this actually helps us to come to a complete um, uh, segmentation that's then usable for layer fMRI. Now, Laney then would proceed by computing cortical rims based on the segmentation, then compute a metric image, which is denotes the cortical depth normalized to values between 0 and 1, and perhaps compute columns, which is optional. Now let's talk about the metric images. So when I first looked into this, I felt that perhaps one should use distance transforms. Distance transforms are also a very old image analysis technique going back to the 1980s even. And the advantage is that they actually closely approximate Euclidean distances. So if you did an approximation like on the right-hand side of the screen here using square root of 2 and adding them up, you might actually overestimate distances. And with distance transform, you avoid such overestimations. So here on the left-hand side, you can see what happens if you just simply naively um, add values like ones if you a value of 1 if you go one step in the left or right, and then add square root of 2 if you go diagonal, and so forth. So the blue line would indicate the overestimated length. And I was kind of worried that perhaps if one did that, one might make a mistake. Therefore, I'm using a distance transform to implement the metric image. And I get two distance transforms, one from white matter to CSF and one from CSF to white matter, and normalize it to values between 0 and 1. But then it turns out it doesn't really matter very much. So I compared the output of Leni and my own output, and they're almost identical. Not quite. I think Leni is a bit smoother, but it doesn't seem to make much of a difference. So this would be a full metric image after the post-hoc correction of the segmentation and my distance transforms. Now, what's more important is in Leni, people normally use columns, and they are non-overlapping and based on Delaunay or Voronoi triangulations. And the real difference to my approach is that I use overlapping cylinders. So it's not a parcellation, it's actually overlapping cylinders. And that's kind of the main difference here. So here's an illustration of what these cylinders might look like. So on the left, you can see two such cylinders, just as an example. So each cylinder has a center line, and then it has a radius around it that is user specified. On the right-hand side of the screen, you can see the amount of overlap. So in this particular case, there's values of overlap between 1 and 44. 44 meaning at this voxel, at the dark red voxels, there are 44 cylinders that overlap in this particular voxel. So we have quite a substantial overlap between these cylinders. Now, to identify these cylinders, I've developed a software. Maybe I go back one step. So what you have to do in order to identify a cylinder is you start at one side of the rim. So for instance, at the CSF gray matter border, and find the nearest location on the opposite border, and then draw a line, the shortest distance, and then dilate it by the user specified radius. Now the next step then is to do statistics. And the first part of statistics, the easiest way, is to simply parcellate the cortex into three slabs: deep, middle, superficial. It's something that many people do. One can also use more of these slabs, but here let's talk about these three. And then define those cylinders and let them move across the entire cortex. And in each cylinder, we now do these statistics. So first of all, we do a simple average for each of the three layers. So we get three mean values in this cylinder. And we also do two sample t-tests where we compare the deep minus the middle layer, so the Z-map activation values, and deep minus superficial and middle minus superficial. For the statistics, of course, we have to be a bit careful about the degrees of freedom. So clearly, since we've upsampled and fMRI data are correlated anyway, the degrees of freedom will be much less than the number of voxels in a cylinder. Therefore, in order to still get valid statistics, I propose to do permutation tests. So here in this case, I've implemented to randomly permute depth labels, which will give us a null distribution. I've also checked that the exchangeability under the null hypothesis is respected. That's very important for permutation tests to be valid. Alternatively, one could also use GLM. So here, for instance, I propose to use three basis functions, one basis function for each of the three layers, and they overlap in a way. But the advantage could be that it's kind of removes noise, so it actually only fits regions where there's actually an activation. And the you can see the GLM as it's set up here. So the Y vector would contain the Z values of an activation map in one cylinder. Every whatever I'm talking about now is just restricted to one single cylinder. The X is an n times 3 design matrix, where n is the number of voxels in the cylinder, and beta are the coefficients that are estimated. And again, in order to get valid two-sample t-tests, we can do permutation testing by randomly permuting depth values. And then we would have to recalculate the GLM in each permutation, 1,000 permutations or so. So to summarize, for every cylinder, we get 6 results. So 3 mean values for each of the 3 layers. If we use a GLM, we get beta values, beta 0, beta 1, and beta 2. And we also get Z values resulting from t-tests. Also three such values, deep minus middle, deep minus superficial, and middle minus superficial. The remaining ones are, of course, simply an inversion of the sign. So if you have deep minus middle, then you easily get middle minus deep. It's just the reversed, sign reversed Z value. But that's just within one single cylinder. What we would actually like to have is to get a map of laminar activations. And in order to get that, we simply average across all the cylinders that overlap in a single voxel. So for every voxel in the gray matter, I look at the cylinders that actually hit this particular voxel and average those 6 results that I showed earlier. So these six statistics. They can be averaged because Z values are normally distributed and so are mean values. So it should not be a problem to simply average them. So that gives me a complete statistics in every voxel of the cortex. And once we have that, we can do various types of statistical analysis, for instance, conjunction analyses, and all sorts of funny stuff that we would like to do. But of course, it's still not corrected for multiple comparisons. That's also a bit tricky because we cannot use any of the existing methods for multiple comparison corrections. Because we, for instance, we certainly do not want to impose any Gaussian spatial smoothing. That wouldn't make sense at all. So what I would suggest to do is simply use the false discovery rate as it was proposed by Benjamin Hochberg in 1995. There's this famous paper that was cited, I don't know, hundreds or thousands of times, and one can use that. So let's look at what that would look like. So here's a synthetic image. So I simply generated a synthetic Z-map. And on the left-hand side, you can see the original Z-map. It contains Z values between -5 and +5. And on the left-hand side, you would see the interior, so the deepest layer. Then you have in this layer in the middle, you have a middle layer activation and also superficial activation. You can also have an activation in 2 layers or in a combination of those, even in very highly curved parts of the cortex. And on the right-hand side, you can see the result of a statistical analysis using Cylarim. So on the right-hand side, you can see the contrast deep is larger than middle, and deep is also larger than superficial. And the color codes correspond to Z values. So in other words, on the left-hand side of the screen of this image, I don't know if you can see my mouse cursor. If not, just look at the left side. You can see there's bright colors indicating a very high statistical significance for this particular contrast. And that coincides with a Z map. And the bright blue values indicate the reverse, where deep is certainly not larger than middle, and it's also not larger than superficial, which also corresponds to the Z map. Now let's look at another contrast. This would be superficial larger than deep and superficial larger than middle. So what I'm doing here is actually this and sign in the middle is actually a conjunction. So I have two contrasts that I computed in the previous step, and I'm now doing a conjunction, meaning I take the minimum of the two Z values that were previously computed by the permutation test. And again, you can see that it picks the right colors here to to indicate places where superficial is really the largest activation. There's another issue, which is equivolume layering. So it was first noted by Bock in almost 100 years ago now. And there was a paper 2014 by my former colleagues in Leipzig where this was especially addressed. And here I propose to use exactly this idea of using overlapping cylinders. Because the nice thing about this overlap is that areas of high curvature are actually indicated by high values of overlap. So here in the image, you can see the regions of high curvature actually show up bright red. Those are high curvature images, high curvature parts of the image, and they indicate high curvature. So the principal idea is that for every cylinder, we get a histogram of depth values, and we can weight them by the overlap count, and then use histogram equalization to make distributions of depth values uniform. And that's really what one would like to have in equivolume corrections. So here's the basic idea. So for every cylinder, we calculate a weighted histogram of equidistant depth values. And voxels with lower cylinder coverage receive higher weights in the histogram. The weight for each voxel is determined as 1 minus the cylinder percentage at that location. And then we apply histogram equalization. What histogram equalization effectively does in this case is that cortical depth then obtains an approximate uniform distribution in every cylinder. In other words, this histogram equalization ensures that each layer defined by the adjusted depth values contains an equal number of voxels. And I guess that pretty much covers what equivolume layering actually means. So here's a comparison of the results. So equidistance on the left, Cylarim in the middle, and Laney on the right. And you can see that Laney is a bit more extreme in this equivolume layering. Cylarim is a little bit, well, less pronounced, but it goes in a similar direction. But I guess, of course, there is an issue with segmentation noise, and effectively all algorithms are wrong. I'm not sure if Renzo produced this image, but it could very well be. And I must admit that I haven't really tested in how if my algorithm is more stable than other algorithms. I'm really not sure if that's the case. But in principle, equivolume layering amplifies discretization noise. Any segmentation error gets amplified. Now let's look at results. So I'm still using data from the Leni test site. So this is VASO data. In the bottom of the screen, you can see the where the test data came from. Here I've used a cylinder radius of 2 mm. That leads to a mean cylinder size of 2060 voxels approximately. Of course, not all the cylinders have the same size. Depends also on the curvature and other factors. And you can see the deep, middle, and superficial results. So deep meaning those are areas where the deepest layer is actually larger than the middle and the superficial. So it's always a conjunction of these two contrasts. And one can see that the middle layer is actually has a pronounced result in the center of the screen. That's where the activation in the middle layer is clearly stronger than that in the other layers. But even in the deep and superficial layers, you do find areas where you do have a layer-specific result. I should add that in this case, I haven't used equivolume correction at all. This is just equidistant correction. I don't know if that would perhaps make a difference. Perhaps it would. I'm not quite sure. Here's the result if I use a slightly larger radius. This would be 3 mm. The mean cylinder size is then almost 4000 voxels. And now you can see that the um, middle activation or the activation of the middle layer becomes more prominent here. So the image is more clearer here, and perhaps it kind of indicates that perhaps the 2 mm result was too noisy. Let me go back again. This is the 2 mm result, and this is the 3 mm result. So clearly, it helps to have slightly larger cylinders. This is the same result if I'm not using a GLM, but simply the means of the layers. And here I'm just looking at the middle layer, not at the deep or superficial layers. On the left-hand side, you just see the mean of the middle layer, and clearly this middle layer activation comes out quite prominently. In the center of this slide, you can see the two-sample t-test, the conjunction analysis, and that is much stronger than for the GLM. I'm not quite sure why. I suppose there's still a lot of noise in the data. Perhaps one needs to go for even larger cylinder radii. Here I've used 3 mm again. And then, of course, one can do an FTR correction. Now we are actually doing 3 such tests, so it would be better to have a correction factor, not just alpha of 0.05, but I'm a bit more conservative here. But still, you can see that almost everything here is statistically significant. Perhaps someone has an idea of why this might be. Let's also look at an experiment we did here in Tübingen, which is at our 9.4 MRI scanner. So we had 9 subjects and 2 sessions each. The anatomy was 0.6 mm cubic. We had segmentations done by Julius Steiglechner into gray matter, white matter, and CSF, which are then subjected to this post-hoc correction. We have functional data, BSSFP and EPI of 0.8 mm cubic, and all of that was scanned at our 9.4 scanner here at the MPI in Tübingen. The experimental design was moving checkerboards. Subjects were instructed to count the number of up and down movements of these checkerboards. There was one condition where these checkerboards had a high contrast or low contrast. And there was another condition where subjects were instructed to either pay attention to the left or the right side of the screen. But I don't want to give a full result here because we're still in the process of analyzing the data. So here's one subject and the BSSFP um, sequence. This is the anatomy, upsampled to zero point 0.2 millimeter. This is the classical Z map, which is a contrast between high and low. And you can clearly see there's a lot of activation in the visual cortex. And this would be a laminar map. So from uh, previous studies, we would assume that in V1 we would have a stronger activation in the middle layer compared to the other two layers. And that's actually what we can see here. But also notice that it's not everywhere in V1. There are also some areas in V1 where we do not see this, which also would indicate that just going for atlas-based region of interest, we will probably not get a good statistical result. Yeah, finally, let me talk about the software. So as Renzo said, everything is implemented as part of the Lipsia package. Lipsia is an old package that I started developing with my group in the Max Planck Institute in Leipzig many, many years ago. And it's now has its new home in Tübingen at the MPI. It's developed on Linux in C, and um, it's relatively easy to install because it's a small package and everything is a standard C. One can also use Singularity or Docker containers to let it run on other hardware, other operating systems. And there's a GitHub page that you can check out for instructions. So let me show you how to use this. Let me first say that these post hoc corrections of the segmentations that I talked about initially haven't yet included them. The reason is that I've only tested them on our own data, so 9 data sets measured at 9.4 Tesla. So if any one of you is interested in this particular part of my talk, then uh, perhaps one can discuss that, and I would certainly need more um, training data and check it out more clearly if it's actually a robust algorithm. It was quite robust in our data, but it's just these 9 data sets all measured on the same scanner with the same sequences. The software is compatible with Leni. So if you do have RIM or metric images that were produced by Leni, they can be used. But there are some constraints. So the RIM image must be an integer, character, unsigned character, and it must have standard coding. So the color codes need to be a 3 for the gray matter interior, need to be 1 for the gray matter CSF border, and 2 for the gray matter white matter border. I think that's kind of standard, but perhaps not always. And the metric image can also be used if it has been produced by Leni. Also, if there has been an equivolume correction, you can use that, but it must be in float or double, so 4-byte or 8-byte floating point, and it must be normalized to values between 0 and 1. So if it's in Nifty, you first need to convert it to our format, and that's what the next 2 lines show you. So you just say vNifty, and then input is the RIM image that you got from Leni, perhaps, and you get an output image. We have our own software, our own data format, which is called Vista. So our data have the appendix.v. And you can also do the same with the metric image. So that should be quite easy. If you do not have these images available through Leni, you can also use them from Cylarim. So you do call a program from Cylarim, which is called vrim. So all the programs that come from Lipsia begin with a V because of the Vista format. you need segmentation as input, and you get a rim image as output, and so that's kind of straightforward. But more interesting is what you do with the Cylarim itself. So the cylinder segmentation and definitions. So you need a Z map, so an activation map somehow. The metric image, the rim image, you need to specify the radius of the cylinders in millimeters. So here in this case, it would be 2 millimeters. You need to indicate the type of statistics you want to use. That would be GLM or slabs, which is simply the averaging. The number of permutations if you want to do permutation tests, and then you get an intermediate output image. For instance, might call it cyl.v for cylinders, and that just contains all the information you would need for later statistical analysis. And the statistical analysis is what follows. So uh, you can then call another program which takes the output of v Cylarim as input and then does various statistical tests. So for instance, if you and you specify the test you want to look at by the minus type parameter. So if you do uh, 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 minus type Z deep, it would tell the program to do a conjunction analysis where the deep layer is contrasted against the middle layer and also as a conjunction against the superficial layer. So that's would implement a conjunction. There are also other types of tests implemented. So the idea is that it's kind of computationally intensive, especially if you do not have any region of interest. If you want to compute these cylinders across a large part of the cortex, then it might actually take quite a while. And therefore, it's nice to have them in a fixed file and then later do various types of statistical tests on them. Yeah, I guess that's it. And I would like to thank my collaborators, Lucas, who does a lot on the GitHub page, and Julius for the segmentation, and Pablo and Edita worked a lot on the 9 Tesla fMRI data, Andreas, Markus, and Klaus Scheffler, of course, as supervisors. And I did have some funding from the DFG. Yeah, thank you. That's it for the moment.
Thank you, Gaby.
Mhm.
There was a lot of content. How do I start? Maybe I have one question first, where I would like to bring it together with kind of not needing an ROI. So I really appreciate these tools that you can kind of basically 3D browse the data based on areas that have certain layer signatures in there. Usually when kind of what you showed in your quote unquote standard layer fMRI pipeline, there is some randomness in kind of how people select their ROI, because they need to select basically an activated area in V1 or somewhere, and then move in there to look at the layer profiles and then see how they are modulated. So in the Cylarim, it kind of allows you to basically look at that in a 3D space, but still it doesn't really tell you where the border of the ROI is, right? You would need some kind of threshold at the end to kind of extract the data and then look at the layers from there. Or did I understand that right? Or are you saying that basically
Actually, it depends on what you want to do. So one has to be careful that one doesn't get into a circularity. So one shouldn't use the output of Cylarim to define a region of interest post hoc, because that would become logically circular, because then you are guaranteed to get a result. Uh, so that would be like double dipping, effectively.
right?
It's right. So what you can do, I also have one program where you don't do any layer statistics, but just show cylinders with a maximum Z value anywhere in the cylinder. That can be used for defining a region of interest. But that would not be what I've shown here. So this double dipping issue could become problematic.
And I believe there are different subdisciplines in layer-dependent fMRI. I think most of the application folks kind of use the approach that you might have implied in the letter. Like you just look at a maximum set anywhere, and then you take the entire patch of the cortex or the entire column for that matter. But then still you cannot say, oh, look, I have activity there, but then you need to go at the next step and look at the different layer modulations in there. Otherwise, it's still circular, right?
Right, you have to be careful. So to avoid double dipping, yeah. It's actually, you wouldn't really need to do it because it's actually a full statistics.
Are there any burning questions? Otherwise, yes,
I have one, Gabriel. Thanks for a nice talk. Very interesting. Regarding mapping, I mean, in the end, you map the value of the statistics to whole gray matter rim, the place you're interested. In the end, you're doing statistics on the average time courses, that is on the upper, middle, and lower. And actually, there are voxels within those segments that are driving the mean time courses. So I wonder, like, if you can further work on this statistical value to give some weights and get a voxel-wise map instead of having a color for like a large area just indicates if it's feed forward, feedback, or like upper, lower.
That's right. So what I've done right now, so what you're saying is I'm kind of restricting everything to these three layers basically. And one could be more precise. One could even do kind of multivariate approaches and look for patterns in activations. That would definitely be possible. I haven't implemented that. But in principle, it would be possible, I guess.
Yeah, I mean, one can still keep deep, middle, and upper, but you know, there are voxels that contribute to deep more than the others. There's an homogeneity pattern within the deep and upper and lower.
Yeah. Mm.
That could
That's
contribute
right, yeah.
Yeah.
That's right, yes, one could do that. Right.
So, yes. Thanks so much for the talk. It was great. And I really appreciate few points of your talk because I found some similarity in what I've been studying before. So you mentioned at some point that one of the limitations of Laney as a general software is to look at the columns that are not overlapping. And I'm assuming that in that sense you were referring to the program LN2 columns. However, we also developed an extension of it that doesn't have any more non-overlapping columns. That's called LN2 UVD filter that allows you to create such a cylinder as you do for each voxel location.
Ah, is it part of
like-
the Laney package or is it something
Yes,
else?
it's part of the Laney package and it's called the LN2 UVD filter.
Uh-huh.
And so basically you have a complete parametrization of your 3D region that can be just defined. You can also extend it to the whole brain. Don't take it as a 3D, you already like define your region. No, it's like an extended patch. And then within this parametrization, you can choose which feature you want to highlight. You can create cylinder that they have full depth in each location of your voxel. Or you can also have pancakes like, for instance. So it's very flexible. And I did work on that for my project on cortical columns, where similarly to you, I was taking it as input my activation map from a GLM, and I was simply try to understand how columnar was my data as a feature. So I found it being a bit of a cuisine, a sister of what you did, that you talk about a laminarity index, or more or less, and I talked about a columnarity index. But the principle is basically the same.
Oh, yeah,
With the
interesting.
difference that I can like parameterize both the radius and the height of my cylinder. And then I write back whatever metrics you compute within a cylinder in the voxel.
Ah,
for each
interesting.
voxel. I
Uh-huh.
think what you did there is to write back in the entire columns. That's right.
Yes, that's
Yeah,
right. Yes, in the
yeah,
entire cylinder. Yes.
in the entire cylinder. So you are, yeah, in my case, or in our case, we write back in each voxel.
Mm,
And
interesting.
it gave you slightly more, I would say, subtle difference on that.
Okay, I was primarily what I spent a lot of time in was these permutation tests. I was
Yeah,
worried about the
yeah.
uh, degrees of freedom and so forth.
And indeed, and that
Mm,
part that is different from like my approach or our approach in
mm,
general. I didn't do an extra statistics on it, and I
mm,
found interesting that you had this bigger cylinder, of course, because you want to have more voxel inside to do statistics. So at that point, I was thinking about what about can you have as input instead of having your activation map that comes already from a GLM or like some
mm.
kind of statistic, time course, so that then you fully could exploit the power of your methods that is pulling together voxel within like cortical depth, but in a smaller patch, right? With the entire time course. So that then you try to reduce the amount of noise in your timing, and then you do the fitting over there. Still,
So I mean, that would mean so you're saying I could do the instead of doing a pre-processing basically for an
yeah,
activate get an
because
activation map, I could
yeah,
do the activation map as
exactly.
part of this cylindrical approach.
Yes.
Is that what you were doing or?
No, I didn't do it because
Mm,
for me, the property that I want to highlight was this columnarity.
mm.
How much columnar is my resultant map.
Ah,
However, when I heard, like I listened to your talk very carefully, I thought that one big benefit would be actually to do a kind of denoising to the time course, because most of the time the issue that we are having is obtaining such an activation map. So maybe
right.
like pulling together voxel in that smart way will boost your signal to begin with, and the fit will be, of course, like better. And then, I mean, it's just a matter of quantification. Then you want to quantify how different it is within like a columns, like within a cylinder, whatever, but in your region. That will be like a second step that will not require you to do extra statistics.
Yes, that's
Okay.
interesting thing. So both of what the previous speaker and also what you are saying is one could be a lot more, uh, well, sophisticated in what one does statistically within a cylinder, and
Yeah.
that's certainly true.
Yeah.
And yeah, that's a good suggestion. And but it would probably take a lot of computation time, I guess. But it's certainly possible.
Yeah. I think that's a very
Yeah,
good point.
yeah. Yeah. And your cylinders are much smaller, I take it, or
Like in the program in Laney,
yeah.
in
Mm,
UVD filter, you can choose your
ah,
radius and your height. In my
mm,
case, in my study at point 0.8, I choose to have a 0.4 as a radius because I
okay.
was looking for very small
You're actually looking for
local.
columns. Yeah,
Yeah,
yeah.
exactly. I was
So
looking for
I'm
columns.
yeah,
I need that
I
specific
see.
resolution, I would
Okay.
say, nominal
Yeah,
resolution.
I wasn't thinking of columns. I thought columns are beyond our reach in fMRI, but yeah. Okay, I understand. Thank you.
It's a quantification. It's a
Mm,
data-driven way of saying, I have a map. Does it look like columns
mm,
inside? And I
mm.
think it's similar to say, okay, are there some laminar pattern in the other way?
Right. Thank you.
Yeah, thanks to you. It was very inspiring. Then I also would like to highlight, if I can, one more things about your segmentation and the way that you were polishing it
Mm,
afterwards using a morphological operation. There is
yes.
actually another program in Leni
Ah.
that is based on a similar concept that's called LN to rim polish.
Ah, yeah. So it does
And
something also a
yes,
morphological filter, or does it?
yes, exactly.
Uh, so
So it does a combination of erosion and dilation Then try to remove mostly also the noise that you can induce if you do manual correction.
Does it do a gray value morphology or binary morphology?
Again, sorry?
Does it do grayscale morphology or binary morphology?
Binary. It
Binary,
starts
yeah.
with binary, and then it does some Gaussian operation on it, and then it add morphological operation, dilation and erosion. Again, a Gaussian smoothing, and then binarization.
Yeah. So
So even
here's
if there is a binary input and a binary output in between, of course, it has to be smoothed out. And
okay, so here the trick is actually to go to the grayscale image and do grayscale morphology.
yeah,
That's the trick.
I think it will be very interesting to combine them,
Yeah.
like to see after you use that operation, because I saw that at some point, indeed, it was like not continuous and stuff. But if you would use such a tool afterwards, maybe we can reach a further,
Perhaps.
less manual work.
Yeah, let me know.
Yeah.
Great, thanks.
All right, thanks. Yeah.
I have another question. I found your way of doing equivolume really cool. It's inspiring, and it might be a solution to like much further issues that I sometimes see, like if by looking at the overlap of the different cylinders that you had, you kind of got your equivolume field. And one problem that I'm struggling, or maybe others as well, is that we don't really know the shape of like columns or the shape of borders, so to speak, right? When you look at very, very high resolution ex vivo big brain data, or even very high resolution multi-echo GRE, you see that sometimes the veins are bent, the columns are bent in the like in the big brain. But there's no real, like, while we have the equivolume, it says volume, like same number of voxels, but volume of what, right? What's the distance, right? A volume definition needs a size of the column. And if the columns are not straight, but bent, kind of, you can do anything, right?
No.
And then, so I think your approach inherently has a kind of warp field of those. So maybe not only going to equivolume layering, but then you can even use that information with the warp field that you have, that you use for your equivolume column, like layers, then to also get the columnar shape. Do you think that's expandable in that way?
Would be. I mean, there is some dependence on a parameter in my case as well, because I need to define the size of the cylinders, and the amount of overlap depends on how big the cylinders are.
But wouldn't it just scale everything if your cylinders are?
Yeah.
The field would look the same. It's just different scale.
Right, could be. I haven't really tested it very closely. I was still kind of skeptical about what I'm doing, but yeah, it could be. So let's discuss that perhaps in 2 weeks' time in Annapolis.
There is a kind of a minimum kind of column size for you, right? Like you have a lot of at least 6 statistical parameters, right? So if you're making a column too thin, it will be rank deficient quite quickly, right? Which
yeah, but I mean, if you upscale, then you quickly come to dozens of voxels. So it's usually if it's just one line, so you can actually do a line or one voxel thick line that connects the two parts of the rims. That would certainly not be enough. But just having one layer of dilation of one voxel is probably enough to make it not rank deficient. Also, one could do it twice. One could have compute these cylinders just for the purpose of equivolume correction, and then do it again for the statistics, the laminar statistics, and then use larger cylinders.
Yeah, no other questions. I would like to kind of mention one more thing. In the very early versions of Laney, the distance metric that you talked about gave me so much headache, and specifically because we have this discretization, and then kind of the cortex is not always the same thickness, and finding the closest voxel on the other side is quite a challenge. and so kind of using this flooding or like growing versus erosion operations step by step, by voxel by voxel by voxel, to find the closest voxel was a huge leap forward to make the layer smooth and not jagged. But then, however, as soon as you found the closest voxel, then Laney does use the distance after all, which might be why the results were so similar. It's just kind of the hard challenge of finding the closest voxel, which is the square root of 2 going over and over again. And as soon as you found it, then you can take the distance.
I see. But you don't have to do that because there are these distance transforms, and I'm using what's called the constraint distance transform. And the constraint is that it shouldn't walk across white matter, for instance. It should stay within gray matter. And there are special algorithms for that, and I've implemented them years ago, and I simply use them. So the metric code is very small in my case. It's just two calls to the constraint distance transform, basically. So you don't really have to find it. So these algorithms work by moving kind of a pattern across the image twice. There's a backward and a forward and a backward pass, and that's all you need. So you don't really need to find the opposite rim. It's automatic in the algorithm.
Well, in real data, very often kind of the closest voxel of the opposite rim is not the one you're interested in, because then you have these chunks of I'm not understanding you. Sometimes then you have kind of at the top white matter voxel of a gyrus then kind of is overrepresented, and specifically if one side of the gyrus is thicker than the other side of the gyrus, or sulcus, for example.
I know.
Then things get very weird. So this
you don't have to do that really. So not for getting the metric image, I struggled with that a lot when I implemented the algorithm for defining cylinders. There I had exactly this problem, and the way I'm solving it is that I'm doing solving this problem twice. So I first get the closest voxel from the outer rim to the inner rim, and then also back again. And then I have lots and lots of overlapping cylinders. And then I kind of avoid this problem, but I know exactly what you're talking about. I had a lot of headaches about that as well. It's not trivial.
It sounds so trivial. It should be easier.
It should be trivial, and I got extremely frustrated by it. But yeah. And in the end, I think there's a way to work it out.
We are at the full hour. Is there any more question? If not, then let's thank Gaby again. And
thank you.
Soon.
Yeah, hope to see you soon in Annapolis.